So this lesson is about transcription. And to start off with transcription, remember that this is where we are trying to get the genetic information that is in DNA, which is housed in the nucleus, out into the cytoplasm. So it is the transfer of genetic information from DNA to a substance that we call RNA, particularly mRNA, which stands for messenger RNA. Alright, and so the players involved are DNA, there is RNA, we have helicase, and we have an RNA polymerase. And just as a couple of reminders, remember RNA is double-stranded, so I'm going to unwind some of our RNA. And let's say we have T, A, C, G, A on this side. That means that our complementary strand is going to be A, T, G, C, T. And we need to somehow get that information out into the cytoplasm. First of all, it's too important for the DNA to be able to leave, because we want to protect that, because that pretty much is all the information the cell needs to survive, and you don't want to destroy that information. And secondly, our DNA is just too fat to actually get out of the nucleus. So instead, mRNA strand is made. And essentially, the mRNA is going to have this exact same code, but in its own sort of version of it. So mRNA, if you remember correctly, it's single-stranded. Let's say this is my mRNA strand here. And it actually transcribes that code in its own language. So a T still will have bind together with an A. We call that a complementary base pair. And the complementary base pair of an adenine in mRNA is U, a uracil. The complementary base pair of cytosine is guanine. The complementary base pair of guanine is cytosine. And then again, for adenine, it's uracil. So you notice that the mRNA's code is actually absolutely identical to the other side of the DNA. So the mRNA has all the information from the DNA. The only exception is that instead of having a T, we have a U. And this then leaves the nucleus to go out into the cytoplasm. And what I think is the best thing to do is actually see a video of how it does that. So for transcription, that is simply all you need to know. So I'm going to get out of this, this PowerPoint where I can do my writing. And discard. And go on to my film. So let me get that larger. And I think I will let this play nice and loud. I think this probably gives us the best idea of how transcription works.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular.
All right, and that pretty much ends transcription. The next step is translation, and that's more or less getting that information that has been assembled by the mRNA that has now moved out into the cytoplasm and translating that into a protein. And remember, a protein is a chain of amino acids. So essentially we're wanting to create a chain of amino acids. So the characters that are involved in this one are quite a few more. Of course, we have mRNA. We have the ribosome, which is made up of rRNA, amongst other things, has two parts. We also have tRNA, RNA. We have, and this stands for transfer, RNA, the T, and we have amino acids. And we're going to also include words such as codon and anticodon. So essentially what this is that the mRNA is going to move into the ribosome. So here I have my strand of mRNA. It moves into a ribosome, which is both made up of nucleic acids and proteins. So it enters in here. Here's my ribosome. And the ribosome more or less brings these tRNAs, and I'll just symbol them as T's, that are kind of floating around into the mRNA. And these tRNAs actually have amino acids attached to them, and very specific amino acids. So tRNA1 will attach to amino acid 1. tRNA2 will get amino acid number 2, so that this tRNA has a very specific function in that it brings a very specific amino acid to a very specific spot on the mRNA. And I'm going to show you a video that shows exactly how it does that, because I think it's much easier to see it rather than to draw it. Alright, so let's play this video here, which does a good job of that. And I'm going to narrate as well and stop it in a few of the spots.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail.
You don't need to worry about that.
mRNA also contains codons that will encode for specific amino acids.
Okay, so let me talk a little bit about these mRNA codons. These are simply 3 base pairs of mRNA. So it's read in 3 letters, all of the mRNA, and we call those 3 letters codons. So for example, you might have AAC being one codon, GTC being another codon. Oh, and my apologies, I can't use the term T, so it'd be GUC. So mRNA is read only 3 letters at a time, and those 3 letters have a specific amino acid that attached to it, and we call those codons.
A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another
So notice that tRNA is in fact RNA. You see all the C's, the G's, the U's, and the A's, okay?
key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds.
So this is a really important part. As we know that the tRNA has a sequence of RNA molecules. At the very bottom, we have 3 letters, in this case UAC. This is going to be the complementary base pair of the mRNA, which is the codon. Remember I was saying it was read in 3. So in this case it's AUG. And only this tRNA can actually attach onto it. It's the UAC one, because remember, U is complementary base pair to A and C to G. And this tRNA has a specific amino acid that it gets.
The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site.
And you don't need to know about the sites. Simply know the process.
The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon.
So you notice that you have now the second codon being UUU. This then brings in the tRNA that corresponds with it, which is AAA. And you notice that that tRNA now doesn't have a red amino acid, but it has a different amino acid, which is orange.
Codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters.
And just always notice these codons and what we call anticodons, which are from the tRNA. So it's red in codons.
These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter.
And remember this chain that looks like a bead of different colors, that's your protein that's made.
When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and
So now I have the mRNA, and if you notice the mRNA that's on the bottom, that's made up of RNA, it actually translated to create a protein that more or less exactly matches what the mRNA had. So here you have the codon for a red amino acid on the mRNA, and in fact the beginning of the protein or that amino acid is in fact red. So that the order of the colors is exactly the same as the orders of the mRNA. So that way the DNA has directly transcribed and translated its information to build this very unique protein. And this unique protein is the thing that will allow you to be able to roll your tongue, or will allow you not to be able to roll your tongue. It's all the sequence of colors that make all the difference. And that's wrapping up translation and transcription.